So let's build a membrane. Somebody's going to have to remind me where we left off. Where did we leave off? Didn't we talk about cell walls for a while? Did we talk about walls and how they separate things? Yeah. All right. Well, in all cells, there's this thing called a membrane. And a membrane acts as primarily it acts as a separation. So a cell wall is for structure. It's to hold the cell together under times of stress, whether it be a lot of pressure from water in or out. It has, and that's primarily the big thing, is water leaving or coming. A cell membrane, a cell wall is important to keep the cell together, because the cell membrane acts as a separator. A separator into different what? What do walls in... We talked about the different function of walls in our lives, right? In our existence. What do walls do in our house, in our houses?
Keep intruders out.
Support. They hold the ceiling up. We talked about that, right? Holds the ceiling up. But what do they do in your house? Let's think about it. Let's think about if we lived in a one-room shack. That's possible. In fact, there's probably still many people today that live in a one-room shack. They have one door, maybe a window or two, but it's just one room. So think about what that means. That means that because it only has this outer area, no walls, right? No walls, no internal walls. Does that make sense? There, you have, let's do a diagram of the floor plan. That means that your toilet, market T, is right next to your bed, market B, which is next to your kitchen, market K. So you're cooking and eating here, and you're sleeping here, and you go to the bathroom here, maybe your shower is right here.
I see what you're talking about now.
So there's no separation there's no walls, right? There's no separation. Do you agree? So this is a one-room shack. Nothing wrong with that. They call it efficiency. People, and a lot of you may end up staying in a one-room shack, right? Oh, it's SH. I don't know why it's put SCAG.
It's like 35.
Oh, excellent. Pass those out, Wayne. Thank you.
I gotta get back to that.
Let me read. I don't know why I put SCAG. All right, so it's a one-room shack. So now, if you're in most of our homes, we have more than one room, right? So they're bigger homes,
That's a big house.
right? Similar idea, roof, 4 walls, maybe a door, hopefully a door, and some windows maybe a big bay window, right? All right, side door, rear door, back door, door to the basement, who knows, a bunch of different stuff. See, but in this house, you might even have several floors, right? You certainly are probably going to have these separators in here. And you might have, now you'll have a hallway, right? So you'll have a hallway, and there's your rear door here. And here's your back door, your front door, and then maybe this is a living room here, and then this is the study, this is the bedroom, this is the kitchen. So now you got a bathroom. Here's a little, maybe there's a side here is a bathroom. So now you have, every room has its own purpose. Do you agree?
Yeah.
Okay. So those are kind of 2 basic kinds of living places. You can have this idea of a tent or shack where you have everybody living in one space and everything gets done in that one space, bathroom, sleep, eating, right? Or you have this idea of separating the rooms into different components. Which you think, for different functions, which you think is a better way of living? Like, more efficient, more productive?
The big house.
The big house. Why is the big house more productive? Yeah. More things done at the same time, right? it's kind of hard to cook a dinner and poop at the same time, right? Like you have 5 people in the room and one person's cooking dinner and the other person's just literally sitting there dropping troll and going poop.
Oh, thinking about
That just doesn't seem right, does it? Or if, like you're trying to sleep and your brother is
no,
next to you.
all you hear
Like literally all you hear is bloop, and you're like, what's that? And you're trying to sleep. That doesn't work very well, right? Right? I mean, that's why we separate them into different rooms. That's why we like, most people, I'm not saying everybody, most people like rooms, different rooms for different functions, right?
Yeah.
We all agree on that, yes? We could sleep in all of us in the same room, and we could all go to the bathroom in the same room and eat in the same room. We could do that. It's possible. We'd have to time it a little better, and we'd have to have discussions about what's right and what's wrong. We'd have to say, okay, bathroom, everybody turns around. There'd be all kinds of things we could do, right? But it's just doing a lot. Where this is really functional, highly efficient, gets a lot done. You can do a lot of things at the same time. All right, you got these handouts, but hold on to these handouts. As soon as you're done passing those out, could you go help her get the rest of them? Thank you so much. So just so you know, so this is two ways of using these things, these separators. I'm going to use the word separators. Now there's two words we use, and it's very important you keep them separate. These are words that kids confuse all the time because they're only half listening and they only half do the work with their brains. There's the cell wall, and then there's a cell membrane. All right? The cell wall and the cell membrane. Oh, it's too close to the top. The cell wall is structural. It does kind of separate as well, but it's not really about function. The cell membrane is somewhat structural. It does have structural component to it. It does give the cell a little bit of structure. I mean, you can't really argue that it doesn't. So there's a small bit of the function is structural. And everybody understands this word structural, right? Yes? Do you have a confusion? Is there a question about what that word means? It has to do with the shape and and holding it together, right? But the main function of the cell membrane, one of the main functions, is to separate. To separate the space that's into different functions. And I shouldn't say the cell membrane. Let me erase that and say membranes. Membranes in general. Membranes in general, their function is to separate. The cell membrane is like the outer wall of the shack. Do you see this? The outer wall of the shack, or the outer wall of the house. What does the outer wall of the shack or the outer wall of the house do? What does it separate? What is this outer wall? Outside from inside. It's the basic definition of a home, right? It's the basic definition of what is your space, right? If it's your space, it's inside the walls. I mean, yes, you could have yard and etc., driveway, but bottom line, what's inside the walls of your house, that's yours, right? The stuff that's out here, who knows? It could be the bears, it could be the skunks, it could be anyone or anything else's, right? But what's inside this house is yours. We all agree on that, right? For the most part.
Yeah.
Well, that's what happens in a cell. When you're looking at a cell and you're looking at a cell membrane, what you're looking at is this wall, this membrane, not wall, excuse me, I keep mixing that up. I use this analogy, and of course in a house we don't have a membrane, do we? All we have is a wall. So it's slightly different. But this cell membrane is also known as the plasma membrane, the cell membrane or plasma membrane. Two different words, same definition. This thing separates what? The outside from inside. This defines the cell. There it is. You want to know what's it? You know what? Want to know what belongs to the cell? Everything where?
Inside.
Inside the cell membrane. The outside of the cell membrane is made of water, H2O. The solvent is water. Of course, there's a lot of stuff in it. It's not just pure water, right? We all understand that?
Mm-hmm.
It could be acidic, it could be basic, it could have a lot of stuff in it. It depends on where the cell is, what's happening in the cell, what's happening in the body. So the solvent though is generally water in living systems on this planet. The solutes can change depending on the species, depending on what's happening, the function of the cell. The solutes in the bladder there's a lot of urea and salt in the bladder, isn't there? Because what's in the bladder? Does anybody know? Pee. So there's a lot of urea and salt in pee. Urea is your body's waste. Maybe any toxins in your blood might be in that pee as well. Those are solutes that you would find in the cells surrounding the bladder.
That's why you pee out kidney stones.
Yeah, that's one reason, because you'll have minerals that are dissolved in the urea that form stones. So the cells of your blood vessels would have blood in it around them, right? The solutes in your bloodstream would be iron and minerals, and you would have urea. There'd be a little bit of urea, not as concentrated as the bladder. You'd have, as I said, salt, sugar, right? Fats, proteins, amino acids. Those would all be solutes in the water, in the liquid, the solvent outside the cells that make your blood vessel. Am I making sense? So what is out here, what this stuff out here is made of, all depends on where this cell is and what's a function of this cell. Does that make sense? The inside of the cell is also made of water. It also has water.
Cytoplasm.
And this inside, though, even though it has water, it's a little different in that it has some other stuff that's going on in it. This stuff in here, the stuff that's in there is called the cytoplasm. What's dividing the cytoplasm, the inside from the outside?
The cell wall.
The cell membrane.
Oh.
See, that's what I said. This is why I hate talking about walls, but it's the only real world example we have, right? The cell wall, it doesn't exist in animals. It only exists in plants and fungi and bacteria, right? You remember that? Those things are for structural components, and we talked about what sugars they're made of, right?
Mm-hmm.
You should know that. That's a cell wall. But in a cell membrane or plasma membrane separates the solutes on the outside from what's inside. Inside there are solutes. What is a solvent inside? Water. So there's water inside, water outside. That is a solvent. So the stuff inside and outside, the polarity is going to make a big difference, isn't it? Whether something's polar or non-polar is going to make a big difference. And half your job in a living system is to make sure that you can make all these things soluble in water, because the solvent's made of water. Your body's made of water. You got to figure a way to make all this stuff that's not soluble, it's all of a sudden soluble. So you have to figure a way to do that. Yeah.
So is water and cytoplasm the
Cytoplasm is that's a good question. The cytoplasm is an area. So everything in here is a cytoplasm. That's the space. That's what the space is called. The cytoplasm has water in it, and then of course there's solutes, right? And what the solutes are depends on the cell type. if you're a pancreatic cell, you probably have a lot of insulin and glucagon being made, right? Because that's your job. If you're a muscle cell, you probably have a lot of actin and myosin, a lot of fibers. You probably have a different shape too. If you're a muscle cell, your shape is probably something like this, see? And each one of these is a different cell. And they kind of lay on top. This would be something like a muscle cell. And then, of course, in a muscle cell, you'd have these little kind of strands, muscle strands, right? And all of you that have eaten meat know what it's like, right? You've eaten stringy chicken, right?
Yeah.
Those strings, those are the muscle fibers of in the chicken. Same with, it's like string cheese actually, but of course cheese is not muscle, right? But it's very that stringy stuff in meat is muscle. The tendon is near the bone. So the tendon and the cartilage is all near the bone. The real hard, thick, stiffer stringy stuff, that is a tendon. If you get a tendon though, somebody's not butchering it very well. The meat, the white meat, when you know how you bite into it and sometimes it's stringy, that's muscle. You have the same stuff in your muscles. They are the cables that pull your bones up and down, right? Think of your bones moving up and down. You need how do you do it? You pull a what? A cable. All your muscles, by the way, work by pulling. There's no pushing. It all depends on it's all about which muscles are pulling, right?
So when you push something, your muscles are pulling?
Yeah, it's just different muscles are pulling. You have muscles on the back of your arm and the front of your arm. And when you're pushing, what's happening is you're pulling on the muscles on almost a pulley system. You can think of it as a pulley system. They just keep pulling in one direction, and that forces your bones to move in a specific direction, which you perceive as pushing, right? Are we all good with that? So all muscles do this. All muscles do this. They contract or they relax. Those are the two options, on or off. On or off. All right. So anyways, they have different, notice that they have different shapes. The cells have different shapes, and there's going to be different stuff in the cytoplasm. There's going to be different materials inside. So each cell type is going to have a different material inside. It's going to be surrounded by a bunch of different stuff depending on what it's doing. In your brain cells, are surrounded by cerebral fluid, right? Which is much different from blood. If we opened up your brain, we would not want to see blood in your brain. We'd expect it to see only inside the blood vessels that are in your brain. Does that make sense? Yeah.
So each cell has a cell membrane in our body.
Say that again.
So each cell in our body has a cell membrane.
Absolutely right. Every cell, period. Every cell, if it's a cell, any plant, animal, bacteria, whatever, has a plasma membrane, has this membrane. Are we all good with that?
Mm-hmm.
That's very important. Now, let's talk a little bit about where people get really confused. Bless you. We're going to talk a little bit about later, not now, but I'm going to allude to it. There's a big difference, the biggest difference among cells on this planet, I'm talking about the planet, not just in your body, is this eukaryote, and what's the other one? Prokaryote. The prokaryote is like the shack. It's a single room, meaning it only has one separator, one membrane. Everything that this cell does happens inside that membrane. There's no separation. You know how we have a nucleus and all the different organelles that you're going to have to memorize very soon? This prokaryote doesn't have that. It has a little bit of DNA in the center, kind of circular DNA, a little different from ours. It has little plasmids, little tiny circular DNAs. It has It has little ribosomes all over the place. It has proteins and solvents and solutes in here. And of course, it's made mostly of water inside and outside, just like every other cell on this planet. It also will have a what? What else will it have? A what? It could have a flagellum. Very good. It could have it depends on the species there, but it could have a tail that helps it swim. It might not though. It could be round. It could be a lot of different shapes. But one thing most prokaryotes have is this thing called the cell wall. On the outside of the membrane it has this thing called the cell wall. And remember, it's structural. Remember, this thing's floating around and needs to protect itself from, well, really from issues on the environment, some changes in water, concentration of water. And we're going to talk about osmosis and diffusion very soon. So it has to protect itself, and that cell wall is what helps it protect itself. Of course, there are eukaryotes that have cell walls, and examples of those are fungi, right? The fungus, and the trees, right? So plants, those are examples. But in eukaryotes, you have the cell membrane. So both of them have cell membranes. The interesting thing here is that in eukaryotes, they also have other membranes on the inside. So you have these membranes that define different spaces. They have different spaces. These are all membranes. They're all folded, and so you have things that are like the nucleus, the endoplasmic reticulum. There's a rough endoplasmic reticulum, and there's a smooth endoplasmic reticulum. There's a Golgi body. There's a mitochondria. There's of course ribosomes. There's vacuoles. There's food vacuoles. There's lysosomes. There's all kinds of different all these have what? What do they all have?
Membranes.
Membranes. All these have their own membranes. You see, prokaryotes have no internal membrane-bound organelles. No membrane-bound organelles. That's a prokaryote. So that includes no nucleus, and that's where we get the word prokaryote. No nucleus. And the eukaryote has a nucleus, but also has all these other membranes. And again, just like that shack and house, the eukaryote is much more what? More stable. I guess I say stable's not a bad. It's more efficient. It's more complicated. It can do more things at the same time, right? It can handle changes better than this prokaryote. This prokaryote is much simpler. Yeah.
You said, what don't the prokaryotes have that the eukaryotes?
Can you tell me? Can someone tell me what prokaryotes don't have? What prokaryotes don't have that eukaryotes do? Internal membrane-bound organelles. Prokaryotes do have organelles. They have ribosomes, and they have all kinds of enzymes in here, but they don't have any kind of membrane-bound organelles. So a lot of the membranes you've heard before: chloroplasts, mitochondria, RER, LER, Golgi bodies, lysosomes, etc. Those are all membrane-bound. Yeah. So of course, one of the bigger differences, that's eukaryote, and there you have just about every cell. But then you have some cells that have this cell wall. They're eukaryotes. They have cell walls. There's two kinds, really. And these are defining kingdoms, by the way. We're starting to define kingdoms here. Because these are kingdoms, these are major groups of organisms. Groups of organisms that don't have chloroplasts. No chloroplasts. Does anybody know what a chloroplast does? Yeah.
It helps with photosynthesis.
It does photosynthesis. That's it. That's what allows for photosynthesis to go on. Then you have these things with cell walls. Yes, they do have a cell membrane. They do have a cell membrane. But they also have a cell wall. You see the cell wall, the shape of it's very they tend to have sharp edges. And on the inside of the cell wall, just like in prokaryotes, you have this membrane, this cell membrane. This would be a plant. And the plant has every organelle that an animal does. It just has a few extra. And I shouldn't have made that green, I don't think. I should have made this yellow or something, because the green I want to save for what? What do you think I want to save the green for? Chloroplasts, yeah. I'm going to call it, because chloroplasts are green. And that's where you get chlorophyll and all that. But you do have this blue one. I'm going to go ahead and label it blue. There's your mitochondria. See, they have mitochondria too, because they eat their sugar. That's what mitochondria do. They make energy. They need energy too, so they make energy.
How do you spell that?
So you'll get all the definitions and all the names of each of the organelles later. But the point is, the key point here is that plants have chloroplasts. And they have cell walls. And they have this other thing that helps them regulate water, which is a central vacuole. All right. So the central vacuole, the chloroplast, the cell walls, plants have all that. Animal and the rest of the eukaryotes don't. Yeah.
So a plant can't be eukaryotes if it doesn't have any chloroplasts?
I can't hear you.
I said so a plant can't be eukaryotes if it doesn't have chloroplasts?
No, it is a eukaryote.
But I thought you said it doesn't have any chloroplasts.
A plant has chloroplasts. An animal doesn't, right? But all of these have what? What do they all have? They have membrane-bound organelles. Do you agree?
Mm-hmm.
They have membrane-bound organelles. They have membrane-bound organelles. They have a nucleus, right? So what do we call things that have a nucleus and membrane-bound organelles? Eukaryotes. So these are all eukaryotes. The difference is that they don't have membrane-bound organelles. So all of these have nuclei, nucleolus, Golgi body, endoplasmic reticulum, rough and smooth, mitochondria. These over here, they don't have chloroplasts. No chloroplasts. I hate when I run out of room.
Tell me about it.
No chloroplasts. But they do have cell walls. They have cell walls. They have mitochondria. They have Golgi body. They have lysosomes. What do we call these things?
What do you call what?
So these are producers.
Oh, consumers. No, wait. Wait. Is the second one insects?
Insects are up here. Insects are animals. They just have an external skeleton. Insects are animals. They're arthropods. But these have no chloroplasts. They have cell walls. We know them, and you know them as what? What do you know them as? Fungi. Or fungus, right? So they're the fungi. They're mushrooms and mold and mildew. They have no chloroplasts, so they are not producers. They are not producers, but they do have cell walls. And they have all the stuff that eukaryotes do, because they are eukaryotes. The difference is that they have cell walls, and in fact they act so much like plants that they were actually classified as plants for a long time, until people said, wait a minute, they're not doing photosynthesis. They're not producing their own sugars. They're eating. So they're missing a really key component of producers, and that's that chloroplast. Everybody good with that so far?
Mm-hmm.
So now today, are we good? Because I'm about to go on. Now I'm going to just focus on the membrane, the cell membrane. Are we good? No questions? Yeah, questions?
So the first one was a human cell, right?
All right, good question. So the first one could be a human cell, yes. It could also be a what? Any animal, including insects. It could be a prokaryote. It could be a protist. So this one, there's a lot of kingdoms that fall into this eukaryote. This is actually, I shouldn't have called these kingdoms. These start to define what we call domains. So eukaryote, prokaryote. But among the eukaryote domain, there are animals. That's a kingdom, right? Animalia. Now we have these the plant kingdoms over here, right? Plantalia. Those are producers. And fungi define a third kingdom. So 1 kingdom, 2 kingdoms, 3 kingdoms so far, right? So animals include humans, yes. They include insects. They include tigers, lions, bears, whales, sharks, fish of all kinds, birds, snails, which are not insects, but still, they're arthropods. Slugs, which are neither, cephalopods like squids and octopi, right? I don't know if it's octopi or octopus. I don't even know. Is it octopi? So anyways, the point is that these could be any one of those, okay? These are fungi. These are mushrooms. By the way, the largest creature on the planet is a mushroom. Bigger than a whale.
Oh, it's
3.5 kilometers wide. 3.5 kilometers.
I don't believe you.
And think of them. It's obviously a kilometer smaller than a mile, but if you think about something that's about 3 miles, not quite 3 miles, right? Because it's smaller. But I don't know if you understand what 3.5 kilometers is, but that area, that's how big this fungi is. Oh, yeah.
I think
It's real.
It's skinny.
There's one big giant one in the Michigan area. It goes and it's really old, really old.
Can you eat it? Because I know so much from the
That's a good question. I don't know. I don't know the answer to that. I don't know if anybody's thought of it, but it's most of the mushroom is underneath the ground. It's in the root system. It's one giant beast that lives underneath in the root system in Michigan that goes all the way for 3.5 kilometers around, and every so often you see the mushrooms come out of the ground. So if you walk through a forest in that area, not all mushrooms are like this, but you walk through a forest, you see mushrooms coming up. People think, oh look, some mushrooms over there, some mushrooms over there. Actually, they're the same beast, right? They just have all of them are the mushroom caps, or the extrusions of this giant creature that's underneath the ground. So they're just walking on the back of this monster.
Wait, so there's mushrooms growing?
It's not a monster. But I'm just saying. I just have like this imagining, like one day the beast is going to wake up and eat something. It's not going to do that.
So
it's a mushroom, right?
So
it's a mushroom.
There's mushrooms growing on top of the mushroom.
The mushroom, the thing you call a mushroom is not a mushroom. Not always. It's what it is a protrusion, like your fingernail. Think of you sleeping underneath the ground and your finger comes out of the ground. And somebody walks by and never seen you before, they think that finger is a thing. And they see another finger on the other side come up. And they start thinking that those are all different creatures, when really it's what?
Just you.
It's just one body connected to different fingers, right? So when you see mushroom caps in Michigan, in those woods, those little mushroom caps that come out, they look like different little mushrooms, right? But actually they're all part of the same creature. 3.5 kilometers big. Bigger than the biggest organism on the planet. So you never think of that when you think of mushrooms, right? But that's fungi. Plants obviously can be very big or small. We know plants. We're very familiar with them. And of course animals we're very familiar with as well. All right, anybody else? Yeah.
So you said that plants produce their own sugar and fungi
They eat. Everything else you see on here, these are the only things that make their own food. Well, there's a caveat. There's something else called a chemosynthesizer. We'll talk about that later. There's an exception to that. Photosynthesizers are pretty much the only producers. There are these other little beasties, prokaryotes, that make their own food using chemistry, no light required. All right, a different system. But we all good with this so far? All right, so let's go ahead and take a look at the membrane, and this will lead you to this activity. This activity you have in front of you. You need to start it tonight. You're going to have to use scissors to cut it up. The directions are there if you follow it, and I'll switch over and show you go through the instructions with you. We got 7 minutes, so I have to hurry up. We'll get it later. So here you have the phospholipids, right? And you know that when you have, it turns out that in a membrane, a cell membrane, you know what? Let's not try to do this till tomorrow in class, because I really want to finish a good discussion of the cell membrane before you start trying to do this. I don't want you to not understand as much as you could. So let's continue anyways with this discussion. So you have this, we talked about a micelle, right? Yeah. All right, so micelle is what happens when you put a bunch of these phospholipids with hydrophilic heads and hydrophobic tails, and they all dissolve each other. The tails dissolve each other. Do you remember that? Yeah. All right, and this is a cutout, so it would be a sphere, right? It would be all the way around. And this is what happens with detergents and soaps, right? When you clean soaps, the lipid, the fat from the grease from your French fries goes right into the lipid-soluble section, right? And gets washed away. How does it get washed away? It gets washed away because this part dissolves the lipids, or fats, right? And then this part out here is water-soluble. And so the structure ends up being like a fat carrier. Do you agree? Yes? We're good? All right. It's not as useful as can be because that means that everything in here is fat. Well, how do we make a space that has water-soluble inside, water-soluble outside, but lipid-soluble only in between to keep it, make it a barrier between two sides? Well, what life has done is create these phospholipid bilayers. What does bilayer mean? Two layers. So 2 layers of these, and you can imagine that this would be going all the way around, right? So I'm just going to go ahead and kind of tail it off like this. Can you understand what this means? That when I draw it this way, do you see what I'm saying here? This is just a kind of a close-up. When you're doing this, when you're creating a cell, you need to have that space, that separation. Right? All the way around. Maybe I should use a different color, huh? Too late. You get my point here? You get this bilayer of lipids. Now this is great. That means that if it's not water soluble, it's not going to get in through this barrier, right? This membrane is a good separator. But what do you have to do that this membrane won't let it happen? Won't let happen. So far, I've drawn this membrane, and I've said it acts as a good barrier, right? So if you have something that's soluble, that's polar, right? You have something that's polar or charged, or it's small, you better write that down. I shouldn't hear people putting stuff away because I'm telling you some information is going to be on tomorrow's quiz. It's going to be this. I'm going to give you a quiz tomorrow. I'm telling you what it is. In this, I'm telling you, write it down. If you have this stuff that's soluble in water, how do you get this into the cell? Why won't this get into the cell? First of all, anything that's polar, charged, this is what is soluble in water, right? So this is polar or charged. Here's charge, it's soluble. Why can't it get in? Huh? Cell membrane. It has to get through the lipids. That's right. Good. Somebody's paying attention. It has to get through this lipid section. This lipid section is nonpolar. It won't let it through. It tries to get in here, it won't dissolve. It won't let it pass. That's good. That's what it's there for. Separate it. But you need it to get in here. How do you get it in? So, I mean, think about it. You know the answer. You have this wall here behind me. You want to get in and out. What do you need?
The door.
A door. You need a door and a window, right? How do you create doors and windows in a membrane? That's one of the labs I want to do this week. Is I want you to create a membrane, a door in a membrane. It's going to be kind of cool. You can use we can simulate a membrane using bubbles, like you used to play with as a kid. Those little bubble things. Say that again. Any bubbles, right? Remember when you were a kid, you had those little plastic rings, you'd stick it in a little soapy water and you'd blow a bubble?
And then you can get something small, wet, and put it in.
Yeah, you've done it before? All right, good. So anyways, what we do, when I say we, what life does is it uses these proteins. Remember, proteins are the things that give function to the cell. And it uses proteins that we call integral membrane proteins, and it creates doors. Integral membrane proteins. Integral? Integral means what? To integrate means it's part of. Integral membrane, part of what? Part of the membrane. Proteins. And what's that mean? A polymer of amino acids. Right? A chain of amino acids that fold upon itself. So you got all those different side chains. There's water in the center. There's water here. There's water on the inside. There's water on the outside. What do you think? And we know the heads are all hydrophilic, right? We know the tails are not. So let me ask you, what do you think is happening here? What do you think is going to happen? What do you think is going to happen to these proteins here? Which of these amino acid side chains which you think are going to be hydrophilic? Probably these, right? These are all probably going to be blue. Why are they going to be blue? So they can interact with the water. On the inside, this is a cutout, right? So the inside here, this is all going to be probably blue, right? So these side chains are probably going to be hydrophilic. These are all probably going to be what? These side chains are going to be what? Hydrophilic, lipid-like, right? They're going to have lipid. I don't put things away yet. These are all going to be hydrophilic, right? And then what do you think about this part here? The part that's touching the lipids. This part here. This yellow part. What does it have to be? It has to be nonpolar. So these parts of the protein have to be polar because they're touching water. They're dissolved in water. The parts that are dissolved in the membrane have to be nonpolar. So this protein folds and folds and folds and creates this channel, this hole. So now the sodium can do what?
Open door.
Now the sodium can just go right in. All right? Or out. All right, we'll talk more about these proteins. They're very important. All right, thank you for your time. I hope you learned something today. Yeah? Yeah, I'm going to post 2 videos tonight. You should watch whatever you have missed. So that's how you let things in. Now let me redraw it a different way. If we were looking at it from the top, what we would see is all these. What we would see is all these. This membrane, right? We just see membrane. There'd be a bunch of different heads on it, and I guess I could draw heads this way. So what are these? These are what? The phospholipid heads, right? Do you agree?
Yeah.
So these are all, and then, so, you know what? Maybe I should not draw it that way.
Okay, what if the
What if I draw it this way? What if I'd use blue instead? And so what are we looking at? The heads, right? Everybody okay with that? And we're in water. We're in the water looking down at the surface of the cell. And then we see something that looks like this. And there's this channel in between there. Of course, outside there's water, and inside there's what?
water?
Water. So this is all water out here. There's protein, and of course, there's water on the inside as well. So that this is outside. This is outside, and this is inside. And there's our channel. There's a way in. Does that make sense? And if we looked at it from the side, it would look like this. This part of the protein, this is one big protein, right? Or a few proteins put together, whatever. But this part of the protein here has to be hydrophobic if it's going to be sitting in the membrane. Yeah?
Oh, there's the bathroom.
Yeah. Are we all clear on that? That's one kind of protein. Everybody got a copy of the Build a Membrane, right?
Yeah.
All right. There are a couple other kinds of proteins. And you'll see that there's a receptor protein. Now, if you're a cell in the pancreas, how do you know? You know what a pancreas does? Anybody in here know what a pancreas does? It creates insulin and glucagon. That's right. It helps control the sugar level of your bloodstream. That's correct. So if you're a pancreas, how do you know that if you have to produce insulin or glucagon? How do you know if you need more insulin or less insulin? How does your brain cell know that it's time to tell your body to grow? How does your body know to grow? How do your bones know to stop growing or to keep growing? Or how do you tell your body move? How do you say think or love or hate? How does that work? How does one cell talk to another cell? Cell communication, yeah. And it does it through these things called receptor proteins. So if this is the cell membrane, notice I'm drawing two sides. You see that? Your cell membrane would have this thing that sits out. This is a protein. It would sit out here. They'd have another part of the protein that would sink through this here. And then it'd have another part of the protein that sticks out over here. All right? And this would be called the receptor protein. Now, what's the difference between this and the one we just drew? Is there a channel? Is this a doorway into the cell? No, it doesn't. It's not. There's no way into this cell through this protein. So what this cell does is that when it finds, when this signal,
So it's like an enzyme.
that's right, it's very much like an enzyme. This signal, in fact, some of these are enzymes. Not all of them, but some of them. This signal comes from another cell. Maybe this is sugar in your bloodstream. Maybe this is a hormone, estrogen or testosterone. Or maybe it's a million different signals in your bloodstream that allow your cells to communicate with one another. If this attaches to this, then that changes the shape of the protein. And when the shape changes, when these two match up, when the signal protein matches, and the protein in the inside changes its shape, it does something. What does it do? I don't know. Maybe before it did not bind cyclic AMP, and this is one of those things that maybe this, I don't know. It could do a lot of different things, but let's say there's an AMP. You don't know what that is yet, but let's say it is. It can't do anything with AMP, it's just sitting there. Well, once you change it, once you bind, all of a sudden AMP, usually it's ATP or something like that, gets changed into cyclic AMP. Now this thing, somehow this goes off and sends a signal to another, say there's a whole chemical reaction, a whole bunch of chemical reactions that happen. And that tells the nucleus to make insulin, or make channel proteins for sugar, or it does any number of things it could tell the cell to do. So that's how one cell communicates to the other with these things called receptor proteins. Now let me ask you something. What do you think this part of the protein is that's in the membrane? What do you think it is? Anybody? This part of the protein, what is it? I'll let you answer. I'm going to see if there's any coffee left in my cup. No, okay. Well, why am I saying there's a difference between this protein, this part of the protein, and this part? It's all one protein, and this part of the protein, and I'm saying there's a difference between those and this part that's on the inside. What's the difference? Anybody? Yeah?
Because they're surrounded by different particles.
That's right. So what is this? What must this be? This portion of the protein, what must it be? What must this portion of the protein be? It's sticking in what? In the cell, which means it's sticking into water. So what must it be? Polar. This is where? Inside the membrane, which means it must be what? Nonpolar. And this is what? I hope you see that, because that's going to be a big part of your test, is you understanding what parts are polar, what parts are nonpolar, and how these different proteins work. And I'm not going to ask you to memorize all the different pathways, but just a general idea of how a protein works and what's the difference between them. There's another kind of protein that sits in the membrane again. And it sits in and does this. And it generally actually has, I'm going to give you one that transports using maybe this is, it could be a lot of things, but let's say it has this charged ion here. +1 charge, and then over here there's another one that has +2 charge, two different atoms, right? Ions, molecules, what have you. If this one, and I'm going to go ahead and make this one green, oops, I'm going to make this one green, and I'm going to make this one yellow. If this one comes in and binds here to this green spot, and this one binds to this yellow spot, then what's going to happen is this protein is going to change its shape again. You hear this protein changing shape? This is going to be a theme you're going to see over and over and over and over and over again. Proteins changing shape is a big deal. And what happens when this protein changes shape is that once they bind, right? So the first step is that they have to They have to bind. Once they bind, this thing, the binding of the proteins to their binding sites, this binding, this +1 and the +2, this yellow and this green, once they bind, then they change the shape of the protein, and what happens is that it allows things to come in. It is allowing things to come in, but it's not a channel, right? Do you see what happens? This thing here opened up because it bonded, because they were binding to each other. Jesus, give me strength. So this thing, this +1, this +2, this green thing, oh jeez, this green thing and this yellow thing, when they get inside, they leave. And what do you think happens when they leave? What do you think happens when this green goes through whatever steps, this +1 and this +2, this yellow and this green? What do you think happens to these as they come through? Anybody? Yeah.
The thing close up.
That's right. It goes back to what it was before. So somebody's paying attention. That's good. And it's ready to pick up another one, another 2. And it keeps going round and round. So this is a protein that sits in a membrane. It binds to something. It changes shape. Once it's changed shape, once it's bound, it changes shape. This side closes, this side opens. When this side opens, these no longer are attracted to one another. They get out, they get dropped out. These just float away or get taken away. Once they disconnect, this protein goes back to the way it was and is ready to pick up 2 more. And it does this all day long. Huh? It lets them in. But notice it's different from this one. This one's just open, right? It's just an open door. It's going to let this sodium in all day long, right? A sodium channel. That's what we call a channel protein. This integral membrane protein is a transporter. This one allows things to come in just like the other one did, except that this one does it by changing conformations. We call it shape, changing shape, or changing conformation. Conformation is shape. All right, so these change shape. There are other kinds of proteins that we call tethered proteins. They stick on the outside of the membrane. So here's this, again, thinking of the inside here is what? What's the inside of the membrane? Nonpolar. And outside is going to be polar. Remembering that, there are these proteins that have these lipids that are attached to them. The phospholipids are attached to them. And these lipids are attached to these proteins that sit on the outside of the cell. These things, this would be a good example if you had some sugars on it, would be a good example of your A, B, O blood groups. You've heard of ABO? What blood type you are? That has to do with these cell surface proteins that sit on the outside of your cells. If you have this one, you're A. If you have another one, you're B. If you don't have it all, you're O. If you have an A and B, then you're AB. So they just sit on the outside. They're attached. We call these tethered. Proteins. They're sticking on the outside of the cell. ABO blood group's a good example of that. There's another kind that also they stick on the outside. It's a little bit different. You notice how the reason they call these tethers is there's a lipid, there's a phospholipid that's part of the protein that's sticking right in there. Do you see that? Where in this drawing that I'm about to draw is slightly different. and that there's a protein that sits in here. It sits in there. It doesn't stick into the cell. It just sits on the outside. But the outside is hydrophilic. Then what do you think that this little piece here is? What's this little piece here? It's not polar. So this again, somehow it's identifying the cell. It might be an enzyme that's actually doing a function outside the cell. It could be part of the structure of the cell that holds it together. There are cells, there are proteins. Do you ever wonder how your skin sticks together? Why does one cell of your skin stick to the next cell of your skin? Why does your skin stay together? It's not super glue. They're proteins. Proteins stick together. They anchor each other. This is called an anchor protein. So this protein's anchored to the membrane. And its function can be, there could be a lot of different functions, but they're anchored outside the membrane. So these are the four or five different kinds. Let's go back and look at them. You got one, it's a channel protein. These are two examples, A and B, right? 1A, 1B. Then you have the receptor protein, that's the second example. You have a transporter, that was your third. Right? You have, again, this is part of your receptor protein here, right? This is A and B. This is 2A and 2B. Your tethered protein, which is 4, and your anchored protein, 5. So 5 different kinds of proteins you're going to make during this project. You're only going to have 10 minutes to start, but let me flip to the picture of this stuff, and I'll keep recording even though. Do you see these? Look at the different shapes. Once you're putting them together, you have to ask yourself, when we look at the membrane, I'm going to ask you to choose one color for the phospholipid heads and another color for the phospholipid tail. All right, the hydrophobic and the hydrophilic. This solid line, you're cutting. The dotted line, you fold. If you cut the dotted line, you're going to mess up. Are we clear? So cut the solid line, fold the dotted line. This will be a piece of the membrane you're making. Obviously, these are the heads. The neater you color it, we're going to hang these up. So the neater you cut them, the better it is. I want you to put your name in a marker. Here's what I'm going to say to you very clearly. Write your name on the outside of this somewhere neatly and small before you cut it. Color it before you cut it. I can't tell you how many kids cut it all on and fold it all up and put it together and then try to color it and they end up messing it because it's paper, guys. If you use cardstock, you can photocopy this on cardstock. It might make it better, but I didn't want to go through that trouble. You don't have to. Now here is a channel protein. What do you think the outside of this channel protein should be? It's going to be sticking, this part that's going to be touching the inside of the membrane, the phospholipid layer. What do you think, what should that color be? It should be the same color as these tails. The part that's sticking out of the cell should be whatever color these heads are. Does that make sense? This channel should be the same color as what? The heads or the tails? This is a channel. The heads. Because it's the opening to the outside. Receptor protein, this part here is sticking out. This part here is sticking in. So these should be the same color as the head or the tail? The heads. This should be the same color as the head or the tail? No, heads. This, the middle, the part that's sitting in the membrane should be the same color as what? The tails. All right. Transporter protein. This part's sticking in the membrane, so this part should be what color? Tails. This part's sticking out, so it should be, and this part the same as the heads. The tethered protein, this is obviously the same color as the what? The tails. In fact, they are tails, right? This will be a head. It'll lay on top of the membrane head, right? Because this is the head, these are tails. Then all of this is the same color as the what? Heads. And an anchored protein, this part here sticks in the membrane, so this is the same color as the tails, and this also is the same color as the heads. And then you're ready to, once you've colored them, you should be careful coloring them. I would use a colored pencil. Those colored pencils you're told were part of your supply list. I would use those. I would color lightly. The lighter, the more thoughtful you are of your coloring, the better it's going to be. Stay within the lines. I hate to say that, but it's important because kids don't. And cut them, and try to cut a straight line. Try to cut along. Now, if you want to cut them out in class today, you can cut them out today. But again, I would color them first. If you cut them out, you should put them in an envelope so you don't lose any of the pieces. I'll be grading you based on how neat they are. Is your color coding right? And did you fold them and place them in the correct place? That's what you're going to be graded on. And I will grade you and then tape them to the boards. This will be the first thing we hang up. So you have it. You'll have today, the rest of today, and then tomorrow to work on this. Good luck. Leave me alone. You got 5 minutes. That's enough to color. Leave me alone. 30 seconds less now that you want to argue with me. Now you got a minute less. I spent all that time helping you out. I should have just let you do it on your own. Let you. Yeah.